Hey everyone, welcome back. In our past videos, we have used sliding windows and dictionaries to analyze DNA. But what if you just want to find a functional gene hidden inside a massive string of random genetic letters? So, to be a valid gene, a DNA sequence must have an open reading frame, which is short form ORF. It must start with the start codon, ATG, and it must be followed by triplets of DNA because codons are 3 letters long, right? And it must end with one of the three stop codons, which is TAA, TAG, or TGA. You could write a massive, ugly for loop to check every third letter, track the start positions, and hunt for stops, but that is not the professional way. The professional data scientists don't do that. They use regular expressions or regex for short. Regex is a search function on steroids. Let's open VS Code and build an ORF Finder. Okay, so first we need to import Python's built-in regular expression modules, which is simply called RE. Okay, so yeah, let's do that. Import RE. Right. Then after that, we will take a dummy DNA sequence. Okay, so I have already created one to save our time. Okay, so yeah, so let's call it DNA sequence, and we will use this. Okay, it contains some random letters and two valid hidden genes that we'll be trying to find out. Okay, so now we have to write our regex pattern, right? So a regex pattern is a string of symbols that describe exactly what we are looking for. So it looks a bit like a secret code, so we'll break it down as we go. Okay, so yeah, let's try it out. ORF pattern is equal to uh, R, then okay, gene. Okay, so it starts with an ATG, which is the start codon. Okay, then after that, in square brackets, followed by a group of exactly three letters, which can be A, T, C, or G, right? Then outside this bracket, we use three because it should be followed by a group of exactly three letters. So in regex, it is written like this. Okay. After that, outside one bracket, asterisk, and this group of three can repeat any number of times. So we use a star and a question mark for that. Okay, so star and a question mark, right? Finally, it must be end in a stop codon, right? So yeah, so we write this as TAA or TAG or TGA. Okay, I think that was easy to understand because and the vertical bar means or. Okay, this vertical bar means or. So we are telling that it must end in a stop codon, which is either of these three. Okay, so after putting that all together, it looks something like this. Okay, then finally we can print saying uh, rejects pattern ready. Okay, then we will unleash our gene hunter. So now that we have our highly specific search pattern, we just point it at our DNA sequence using the re.find iteration function. Okay, this function will instantly scan the entire string and pull out every single sequence that matches our strict biological rules. So first of all, we will search the DNA sequence using our pattern. So we will call it found genes is equal to re.find iteration. Okay, that is the function name I just talked about. Then we will use ORF pattern and DNA sequence. Okay, after that we will loop through the results and print them. So print we'll use something like hidden genes found. Okay, make it look a bit grand. Then what we will do here is gene count is equal to 0. For the time being, we'll initialize it at 0. Then we will run a loop for match in found genes. We will add the gene count as 1 whenever a match is found. Then gene sequence is equal to match dot group and gene. this command will extract the exact DNA sequence of the gene. Okay, then we will extract the start and stop position. So let's call it start position is equal to match dot start. Okay, and end position is equal to match dot end. Right. Then finally we can simply print it gene gene count is gene sequence, right? And finally, we can print length is length of gene sequence basis, right? And then position is start position to end position, right? And we can add a new line over here. Now run the code and look at the output. The algorithm found exactly two valid genes. Gene 1 is this one. Okay, let's check it. It starts with ATG, right? The next codons are triple A, triple T, and triple G, and it ends perfectly in TAG. It's exactly 15 bases long, which is a multiple of 3, and the biology checks out, right? Then gene 2 is this one. Starts with ATG, then ends with TGA and 15 bases long. What makes Regex so incredibly powerful is what it ignored. There are other random ATGs and TAs in that string, but because they were not exactly a multiple of 3 letters apart, the Regex algorithm knew they would cause a frame shift mutation. So it completely ignored them. And that is how you use regular expressions in bioinformatics. We just built an open reading frame finder in roughly 5 lines of functional code. So Regex is not just for DNA. You can use it to instantly extract patient names from clinical notes, find phone numbers in messy CSV files, or reformat messy protein ID tags. The code will be available in GitHub. Try adding a massive random DNA sequence into the string and see how fast the Regex engine finds the genes. So hit the like button, subscribe, and I will see you in the next video.